Protein science is one of those fields you start to notice everywhere once you learn its language. The softness of your hair after a shower, the way a virus sneaks into a cell, the fluorescent glow in a microscope image, the way bread rises and beer ferments, a lab test that turns blue when it detects a disease marker. Beneath each is a protein doing a job with quiet precision. Proteins are the workhorses of life, but that undersells them. They're shapeshifters, switches, scaffolds, engines, signal carriers, and sometimes artists, sculpting structures at the nanoscale that rival anything we can build. The promise of protein science is simple and audacious. If you understand how sequence becomes structure, and structure becomes function, you can read the language of life and write in it too. Start with the essential picture. Proteins are chains of amino acids, 20 main building blocks in nature, linked like beads, and then folded into intricate three-dimensional shapes. The order of those amino acids is encoded in DNA, transcribed into RNA, and translated by the ribosome, which is itself a giant protein machine. It helps to think of each amino acid as a letter with personality. Some are oily and shy, preferring the crowd of other hydrophobics. Some are charged and sociable, seeking out partners. Some bend the chain, some stiffen it, some can latch onto metals. When the chain emerges from the ribosome, it's not yet a finished tool. It needs to fold. Folding is where the magic and the physics meet. At first glance, a long polypeptide seems to face a hopeless combinatorial problem. So many ways to fold, so little time. In practice, proteins fold quickly and often reliably because local interactions guide them. The backbone twists into alpha helices and beta sheets, stabilized by hydrogen bonds. Side chains tuck into the interior or spread along the surface depending on their preferences. The hydrophobic effect, water's distaste for exposing nonpolar atoms, drives oily residues inward, creating a core. Salt bridges, van der Waals contacts, and sometimes covalent disulfide bonds refine the fit. The result is a native state, not frozen, but an energy minimum in a landscape full of hills and valleys. The protein breathes and flexes, sampling an ensemble of conformations, but it tends to spend most of its time near the shapes that best suit its job. That job could be almost anything a cell needs. Enzymes catalyze reactions that would otherwise creep along too slowly to support life. They do this by binding substrates, aligning them, acidifying or deprotonating the right spot, forming a temporary covalent bond, or stabilizing a fleeting transition state, tricks that lower the activation energy. If you've heard of Michaelis-Menten kinetics, that's the math describing how an enzyme's speed depends on substrate concentration and on its own parameters, how tightly it binds, Km, how fast it turns over, Kcat, and how those combine into efficiency, Kcat, Km. Beyond the equations is a choreography. Induced fit versus conformational selection, loops that close over a bound substrate, allosteric sites that act like dimmer switches. In allostery, a molecule binds at one site and changes the protein's shape in a way that alters activity at another site, an elegant way to build feedback into a network. Not all proteins are tidy sculptures. Some are intrinsically disordered, lacking a fixed structure until they meet a partner or reach a crowded phase. Disorder isn't failure. It's a feature that allows flexibility, fuzzy binding with multiple partners, and rapid regulation through post-translational modifications. In recent years, you may have heard about biomolecular condensates, droplet-like compartments without membranes where proteins and RNA separate out like oil in water. Low-complexity sequences, multivalent interactions, and weak bonds drive this phase separation, organizing reactions in space and time. It's a beautiful example of how physics at the mesoscale, between the molecule and the organelle, shapes biology. Membrane proteins deserve their own spotlight. They live at the boundary, embedded in lipid bilayers, translating signals, transporting ions and nutrients, and setting electrical gradients that power neurons and muscles. Their architecture reflects their environment: transmembrane helices with hydrophobic exteriors, pores lined with polar residues to shepherd ions, cavities that open and close like gates. G-protein-coupled receptors sample ligands and trigger cascades. Ion channels select for a single type of ion with uncanny precision. Transporters rock back and forth to carry cargo across. Studying these is hard. They're finicky, love lipids, and dislike being yanked into water without help. The field has answered with detergents, nanodiscs that mimic membranes, and new structural methods that can capture these shapes in action. Once a protein folds, the story continues with decoration. Post-translational modifications are the cell's way of writing notes in the margin. Phosphorylation adds a charged phosphate group, often toggling activity or creating a docking site for other proteins. Glycosylation attaches sugar trees that influence stability, trafficking, and immune recognition. Ubiquitination tags proteins for degradation. Acetylation and methylation tune gene expression by modifying histones. Lipidation anchors proteins to membranes. These marks are reversible and combinatorial. The same protein can wear different outfits in different contexts, and the pattern of modifications can be as informative as the sequence itself. When people talk about the proteoform, they mean the specific version of a protein considering its modifications, splicing, and sequence variants, the actual player on the stage, not just the character in the script. If you want to see proteins rather than just imagine them, you enter the realm of structural biology. For decades, X-ray crystallography was the workhorse. You coaxed proteins into crystals, shined X-rays through, and interpreted the pattern to build an atomic model. The challenge was getting crystals at all, especially for flexible or membrane proteins. Nuclear magnetic resonance offered another route, measuring how atomic nuclei resonate to infer distances and angles, excellent for small dynamic proteins in solution. Then came cryo-electron microscopy, which freezes proteins in thin layers of ice, images them with electrons, and reconstructs structures from many noisy snapshots. The resolution revolution in cryo-EM, paired with advances in image processing, opened a floodgate. Suddenly, huge complexes that once seemed beyond reach, ribosomes, ion channels, viral spikes, came into view with atomic detail, sometimes in multiple states that hint at their motions. Parallel to the experimental surge, computation leapt forward. For years, homology modeling let you build a reasonable structure if you had a close relative in the database. Molecular dynamics simulations allowed you to watch atoms jiggle and drift, revealing pathways of motion over nanoseconds to microseconds and, with more power and better algorithms, longer. Machine learning had its moment when deep models trained on known structures began to predict folds from sequence with startling accuracy. Structural predictions became reliable enough to act as a starting point for hypothesis and design, especially when combined with experimental hints. It doesn't make the wet lab obsolete. Details of interfaces, dynamics, and modifications still matter, but it does change the pace and the scope of what's feasible. Of course, before you can study a protein, you need to get your hands on it. That's the quiet craft of expression and purification. You clone the gene into a vector, choose a host, bacteria for speed and simplicity, yeast or insect cells for eukaryotic processing, mammalian cells for complex glycosylation, and decide whether to keep the protein inside, secrete it, or target it to a membrane. You might add a tag, a string of histidines for metal affinity, GST or MBP for solubility and purification, a fluorescent marker for tracking. Then comes lysis, clarification, and chromatography. Affinity capture to grab your tagged protein, ion exchange to exploit charge, size exclusion to separate by shape. If you're wrangling a membrane protein, you'll pick detergents or assemble nanodiscs to keep it comfortable. Quality checks follow. SDS page to see size and purity, mass spectrometry to confirm identity, activity assays to prove it's alive, circular dichroism to glimpse secondary structure, thermal shift or calorimetry to gauge stability. None of this is glamorous, but there's a quiet satisfaction in turning a messy lysate into a sparkling peak on a chromatogram and a single band on a gel and then into a protein that does what it's supposed to do. Assaying function is its own art. For enzymes, you measure product formation over time, watch color changes, track fluorescence, or couple your reaction to another that's easier to monitor. For binders, you measure affinity and kinetics. Isothermal titration calorimetry gives you the thermodynamics in one go. Surface plasmon resonance or biolayer interferometry traces association and dissociation in real time. Fluorescence anisotropy and microscale thermophoresis offer sensitive alternatives in small volumes. For interactions in the cell, you use co-immunoprecipitation, split reporters, FRET, or cross-linking mass spectrometry. For dynamics, you reach for NMR relaxation, hydrogen deuterium exchange, or single-molecule methods that literally watch a molecule flicker between states. Zoom out to the level of the entire proteome, and you get proteomics, the study of all proteins in a system at once. The workhorse here is mass spectrometry. In bottom-up proteomics, you digest proteins into peptides. Trypsin is the classic enzyme, separate them by liquid chromatography, and measure their masses and fragment ions to identify sequences and modifications. You can compare samples label-free or use isotopic labels to track relative amounts across conditions. Phosphoproteomics maps signaling by reading phosphate patterns. Top-down proteomics skips digestion and measures intact proteoforms, powerful when you care about combinations of modifications. Structural proteomics borrows the mass spectrometer for shape questions, using cross-linkers to capture who's close to whom, or using HDX to learn which regions are exposed and flexible. The data is messy and rich, and the bioinformatics to interpret it is a field of its own. With understanding comes the itch to build. Protein engineering answers that itch. Sometimes the goal is to improve a function, make an enzyme tolerate higher temperatures or organic solvents, shift a substrate preference, enhance catalytic efficiency. Directed evolution mimics natural selection in the lab. You generate a library of variants, random mutagenesis, recombination, focused changes, screen or select for the trait you want, and iterate. Rational design takes another path. You look at structure and mechanism and introduce specific mutations to stabilize a loop, create a new hydrogen bond, alter an interface. The most ambitious branch is de novo design, creating proteins from scratch to fold into new shapes and carry out new tasks. Advances here moved from small helical bundles to intricate cages, pores, and enzymes designed to catalyze reactions nature never chose. Between these poles lies domain shuffling, where you combine known building blocks to create chimeras with tailored combinations of binding, signaling, and catalysis. Therapeutics have turned protein science into a very practical craft. Antibodies that neutralize signals or flag cells for destruction. Enzymes that replace a missing function in metabolic diseases. Cytokines tuned to rouse the immune system. Hormones and growth factors engineered for longer half-lives. Fusion proteins that bring two targets together. The details matter painfully here. Glycosylation patterns shape efficacy and immunogenicity. Aggregation can trigger immune reactions and reduce potency. Formulations must protect delicate structures from temperature swings and shear. Manufacturing must be consistent from batch to batch under strict quality control. Even the same antibody backbone can behave differently with subtle changes in the variable loops or Fc region. Engineers tweak the Fc to alter half-life or effector functions, design bispecifics to engage two targets, and conjugate drugs to deliver toxins precisely. It's a place where biology meets process engineering, and success demands respect for both. When proteins go wrong, the consequences are dramatic. Misfolding and aggregation underlie neurodegenerative diseases where proteins clump into amyloid fibrils. Prions take the nightmare further, a misfolded protein that can seed misfolding in others. Even without catastrophes, minor variants in sequence can alter stability and function enough to cause disease. Interpreting those variants is part of the modern clinical puzzle, and protein science helps by linking sequence change to structure and activity. Deep mutational scanning, where you measure the effects of thousands of mutations in parallel, maps the landscape around a protein's function and reveals which positions are forgiving and which are critical. That knowledge feeds back into design, prediction, and sometimes therapy. If you're new to the field and wondering where to begin, you can do worse than this simple sequence. Pick a protein with a clear role, read its sequence into a multiple sequence alignment to see what nature conserved. Look up its structure or a predicted model, and ask what the shape suggests about the job. Then express and purify it, even a small domain, and run a basic activity or binding assay. The act of shepherding a protein from gene to gel to function teaches you how the abstractions become real. Along the way, you'll learn the everyday tools of the trade: how to read chromatograms, how to think about buffers, pH, salt, cofactors, how to prevent proteolysis, how to handle sticky proteins that want to aggregate, how to troubleshoot a misbehaving clone. You'll also learn that a good notebook and careful labeling beat brilliance when you're wrangling many conditions. One of the hidden joys of protein science is how much it rewards both reduction and context. At the atomic level, you delight in a single hydrogen bond that resolves a puzzle. At the network level, you see how that bond changes an enzyme's bias, how that shifts a pathway's flux, how that alters a cell's fate. Proteins rarely act alone. They assemble into complexes with stoichiometries that change over time. They compete for partners. They form feedback loops and feed-forward circuits. They respond to the mechanical properties of their environment, pulling and being pulled. They live in a crowded cytoplasm where diffusion and proximity matter. Modeling these worlds asks you to balance detail and scale, to know when a residue-level insight is necessary and when it's a distraction. The field is moving quickly, and not just at the high end. Affordable gene synthesis, benchtop bioreactors, ready-to-use expression systems, and accessible analysis tools are lowering the barrier to entry. Community databases let you browse sequences and structures, search for domains and motifs, and predict features like disorder, signal peptides, and transmembrane regions. Machine learning tools propose designs, evaluate stability changes, and suggest interfaces. Meanwhile, the best labs keep whiteboards full of hand-drawn cartoons of loops and helices because the heart of the work is still imaginative and tactile. You propose a shape, you test it, you revise. It's worth noting that protein science is a profoundly interdisciplinary craft. You'll borrow thermodynamics to think about stability, kinetics to think about binding and turnover, physics to think about scattering and resonance, chemistry to design reagents and probes, computer science to analyze sequences and simulate motion, and a humanist's patience to tell clear stories about what you found and why it matters. The diversity of questions means there's room for many temperaments. If you love tinkering with instruments, there's a home for you. If you love writing code that finds patterns in oceans of sequences, there's a home for you. If you love the quiet art of making a finicky protein behave, you'll be a hero in more projects than you can count. In the end, an introduction to protein science is an invitation to notice how much of life's drama unfolds through these folded chains. A single amino acid change can make hemoglobin sickle, alter an enzyme's gate and redirect a metabolic stream, shift a receptor's bias and change how a cell reads a hormone. A designed protein can capture a small molecule in polluted water, sense a pathogen, and light up a test strip, or sit on the surface of a virus and blunt its attack. Between these extremes is a daily practice. Read sequences, imagine shapes, test hypotheses, respect the quirks of molecules that don't care what your schedule looks like. The field's humility comes from that last part. You can predict, you can design, you can control many conditions, and still a protein will surprise you. When it does, you get to learn something new about how life builds itself from simple parts into improbable, graceful machines. That's a thrill that doesn't wear off.